Let's take a small interesting topic. This is regarding the special senses. What is so special about these special senses? Let's try to understand some very unique special features of the special senses. In the sensory system, we divide the sensations as general and special. The general senses, they are touch, pain, pressure, temperature, and the special senses are vision, hearing, smell, taste, and equilibrium. Now, 5 special senses, and sometimes it's also said that somebody has a 6th sense. So what's so special about special senses? Why would we refer to them as special senses? Let's see some generalized points first. General sensations, they arise from any part of the body, touch, pain, pressure, temperature. Special senses, they have specialized end organs. For vision, there are eyes; for hearing, there are ears, and these special senses arise from those specialized organs only, unlike the general senses. Second point is there is a specific cranial nerve designated for each special sense. Compare that with general senses, which can arise from any part of the body, and they are transmitted via ascending tracts in the spinal cord. Special senses have cranial nerves designated for them. Second cranial nerve for the vision, 8th cranial nerve, vestibulocochlear nerve for the hearing and equilibrium, and then taste and smell. Now, really, let's see the special features. Let's try to understand very special and unique features of the special senses. First, the olfactory sense. Sense of smell, what's very unique? At least 3-4 features that we must know, which are very unique to olfactory sense. First, neuron is a terminally differentiated tissue, and no new neurons are formed after birth. Neurons do not divide, and they do not form, new neurons are not formed after birth. The only exception is olfactory neuron. Olfactory neuron continues to divide throughout life. That's an exception. Now, what's the reason for this? What could be the logic, explanation behind this? The probable explanation is that the olfactory sense, for the olfactory neuron, its cell body is located very superficially or on the external part of the body, in the nose, and the cell body is subjected to, or this olfactory neuron is subjected to the insult, injury on a moment to moment and minute to minute basis. I mean, inhaled, exhaled air and its friction is going to damage the cell body, and therefore probably olfactory neuron has been bestowed upon with this special capacity that it continues to divide throughout life. New neurons will continue to form throughout life in the case of the olfactory neuron. Otherwise, no new neurons for any other functionality is formed after birth. What forms is synapses. How we grow, our brains continue to grow throughout life and evolve. That's because of the number of synapses that are increasing, not the number of neurons. But there is only one exception, and that's olfactory sense. Second point, uniqueness about the olfactory sense is that all the general and special senses, they have to pass through the thalamus to reach the cortex. Whether it's a general sense like touch, pain, pressure, or a special sense like vision or hearing, they all have to relay in the thalamus first, and then they reach the cortex. The only exception again is the olfactory sense. Now, this is unique to olfaction. What is the implication of this? And does olfactory sense meet the other sensations at any place in the brain or in the CNS? Yes. You see, all sensations are meeting in the thalamus, but olfaction is not. It's not going through the thalamus. But there are two places which have been identified where olfactory sense can meet the other sensations, at least in an indirect manner, by giving the collaterals, and that's in the amygdala and in the neocortex. These two places where olfactory sense meets the other sensations. Have you ever thought that why it happens that in the case of common cold, when we have lost the sense of smell, I mean the sense of smell is not being felt in the common cold, we also are unable to appreciate the taste. Why is that? There's nothing wrong with the taste sensation at that time, yet we are unable to appreciate taste. Why? Because the taste sensation needs this potentiation by giving the collaterals at these two places in the amygdala and in the neocortex by the olfactory sense. Olfaction gives collaterals and potentiates the sense of taste, and therefore if this potentiation is not available, for instance in common cold, olfactory signals are not being sent to the higher centers, then this potentiation of taste also wouldn't be there, and then it will not be appreciated. So that's a sense of that's a unique feature of the olfactory sense. Now let's understand one more feature of the olfactory sense, and that is, here is a sense for which transducer region and spike generator region are located on the same cell. Transducer region and spike generator region are located on the same cell in the case of olfactory sense. Let's try to understand what does it mean. For any other sensation, let's say some mechanical sensation, pressure or vibration, there is a corpuscular cell, corpuscle, which is specifically designated for the sense, and we apply a particular form of energy, and the transduction of that energy occurs at one point on this particular corpuscle or this particular cell, and then spike is generated at the first node of Ranvier. Over here, this is the spike generator region. This is true for most of the other sensations, that the transducer region and the spike generator region are on two different cells. As you can see here, we apply touch to this part. Touch is a mechanical form of energy. It will open the mechanically gated channels and an electrical current will be established. Over here, receptor potential will develop, so transduction has happened, and then when it is of large enough amplitude, it will convert into the spike at the first node of Ranvier. So transducer region and spike generator region are located on two different cells for most of the sensations. In the case of olfaction, it's on the same cell. Transducer region and spike generator region are on the same cell. Here is an olfactory neuron, which is a bipolar neuron, and there are immotile cilia on this olfactory neuron, and the chemotransduction occurs on this immotile cilia, and then the spikes will be propagated through the olfactory neuron. So the point is, the transduction which occurs on those immotile cilia and then spike generation occurs on this neuron. This is a part of the same cell. There are no two different cells. Therefore, again, a unique feature of olfactory sense. Mind you, even in humans, only 1% change in the odorant concentration and we are able to differentiate between the two intensities. Just 1% concentration difference is needed, and then we will be able to identify two different sensations, two different intensities of smell. So in that sense, it may not be as developed as some of the lower animals, but yes, it is strong enough. But the other point is rapidly adapting. Olfactory sense is a very rapid, or rather fairly rapidly adapting sense. I mean, if you enter a room which is having a certain stink or a certain smell, initially you would feel it, and later on you just stop feeling that smell. That's because olfactory sense adapts rapidly. Right, so these are some of the unique features of olfactory sense. Let us understand what's unique about the vision. A unique special feature about the vision is that in the case of all other sensations, there are receptors for every sensation. For every sensory modality, there is a specific receptor, and the receptor is resting under normal circumstances. When we have not applied the stimulus to the receptors will be resting at its resting membrane potential, let's say minus 70 millivolts or so, minus 40, minus 70, it's different, but it's resting. And then when we apply the specific stimulus to the receptor, there is depolarizing potential, a depolarization that develops on the receptor membrane. We call it receptor potential, and this receptor potential, when it is more than 10 millivolts amplitude, it converts into a spike, and then that spike will be propagated through the sensory neuron. So this is the mechanism for all the receptors, for all the general and special senses. Vision is different. Vision is very different from this mechanism. In the case of vision, there are receptors, the rods and cones in the retina. And if we are considering that the stimulus for these rods and cones is the light energy, what should have happened is when the light strikes the retina and falls on these receptors, the receptors should have got depolarized, and then at the synaptic body, the action potentials would be transmitted in the optic nerve fibers. That would have been the normal case. But no, what happens is the opposite. You know, when no light is striking the retina, let's say complete darkness, complete dark, no light, no stimulus to the retina and rods and cones, even then there is a sodium cycle which is going on in the rods. The sodium is thrown out by the sodium potassium pump and it goes back in. So this sodium cycle is going on. This is the dark cycle, the so-called dark cycle, and because of this there is a steady release of neurotransmitter glutamate at the synaptic body. This is in the complete darkness. When light strikes the receptor, what happens is the sodium cycle is disrupted. Now try to understand this, that when the stimulus was not being applied, still there was steady release of neurotransmitter by the receptor, and there were steady impulses in the optic nerve fibers, the sensory nerve that starts from the receptor. By the way, this is called as dark noise. You know, rods are firing asynchronously in the complete dark, and that's called as dark noise. Now, when the light strikes the rod, what happens is the sodium cycle is disrupted, and therefore sodium is leaving the rod but cannot come back, and therefore the rod is hyperpolarized. The rod is hyperpolarized. Sodium is leaving, but it can't come back. Positive charges are going out of the rods. That means excess negativity on the inside, which is hyperpolarization. So rods are hyperpolarized when light strikes the rods. So that's unique and special. For all other sensations, we saw that receptor is resting when the stimulus is not being applied, and then when we apply the stimulus, the receptor is depolarized, and then that gets converted into action potentials in the neuron that is attached to the receptor. It could be any other sensation that behaves like that. We apply stimulus, the receptor depolarizes, and then action potentials are generated in the sensory neuron. But in the case of rod, what we see is already when no stimulus is being applied, still there is steady neurotransmitter release at the synaptic body, and steady impulses are already being sent in the optic nerve fiber. And when the stimulus is applied, light is the stimulus, when it is applied, the rod gets hyperpolarized instead of getting depolarized, and there is a decrease of neurotransmitter release at the synaptic body, and there is a decrease of impulse traffic in the optic nerve fiber, and that is taken as the sense of light. I mean, imagine that in the dark, when the stimulus was not being applied to the receptor, still the receptor was active and it was releasing the glutamate, the neurotransmitter. When the stimulus is applied, its activity is decreasing actually. Neurotransmitter release is decreasing. The rod is getting hyperpolarized. So the point is, the rod receptor potential is a hyperpolarizing potential and not a depolarizing potential like other receptors. It's a hyperpolarizing potential. What could be the reason for this? That it's behaving in an opposite direction as compared to the other receptors. Probably the answer lies in evolution. The humans initially lived in the caves in the complete darkness, and they had developed a certain behavior of their visual system, rods being continuously discharging the neurotransmitter, because dark being the normal, humans living in the caves, and then light became the abnormality probably when they started getting exposed to light. Therefore, probably this is the answer. The answer lies in evolution, that visual sense behaves in the opposite manner. But remember, all receptors get depolarized when stimulus is applied. Visual receptors, they get hyperpolarized when the stimulus, that is light, is applied, and they are already active in the dark when stimulus is not being applied. That's very special and unique. The other senses, what's unique? Let's try to understand the sense of hearing and equilibrium. This is carried by the same nerve, the 8th cranial nerve, the vestibulocochlear nerve. What's unique about it? Let's take the case of hearing. The receptor for hearing and also for the equilibrium is the hair cell in the cochlea and hair cell in the vestibular apparatus. Now, in the case of all other sensations, general and special sensations, how the receptor gets depolarized, how the neurons get depolarized, is that sodium enters into the cell. This is just a generalization, that sodium enters the cell and the receptor or the cell is depolarized. So that's a very common way of depolarization. Or at some places, intracellular potassium from the core of the cell, it just comes near the membrane. That's one more way of depolarization. This also occurs at some of the places in the body. So we have two common ways of depolarization of any cell. Either sodium, the cation, comes in and therefore positive charges are brought in via sodium entry, or potassium, which is intracellularly at high concentration. From the core of the cell, it just comes near the membrane on the inside, and therefore membrane is depolarized, membrane excites. Now let us understand what happens with the hair cell, the receptor for hearing and equilibrium. The hair cell is surrounded by a fluid called as endolymph. Endolymph, the fluid surrounding the hair cell, is having a high concentration of potassium. Endolymph has got a high concentration of potassium, and in that sense endolymph resembles, although it is a part of ECF, but it more resembles intracellular fluid, in spite of the fact that it is ECF, but it is rich in potassium concentration, and therefore it more looks like ICF, intracellular fluid. Now, when sound falls in the inner ear, there is bending of these stereocilia, and or in the case of vestibular apparatus, there would be bending of those stereocilia or hair. At the base of the stereocilia, at the top of the hair cell, there are potassium channels, and this high concentration of potassium on the outside or surrounding the hair cell, the potassium enters the cell, and this causes depolarization of the hair cell. So potassium influx causing depolarization of the cell is a very unique feature of the receptors for hearing and equilibrium. As I mentioned, elsewhere the other mechanisms work, but here potassium influx, because, and as I said, potassium is high on the outside of this cell, which is a very unique feature. Elsewhere potassium is more intracellularly. Here the high potassium in the endolymph surrounding the hair cell, and therefore potassium influx results in depolarization of this cell, and that then would open the calcium channels. Calcium would come in, and the neurotransmitter glutamate would be released at the synapse. This is the 8th cranial nerve fiber. So once the transmitter is released, it will excite these nerve fibers. This is how transduction occurs in the case of hearing and equilibrium. But the point is, potassium influx causes depolarization for this cell. Very, very unique. It doesn't happen anywhere else, just here. The reason, as I mentioned, high concentration of potassium in the endolymph surrounding the hair cell. So these are some of the unique and special features for each of the special sense, and for which they are very, very special and unique.